all that happens, and you just think in your back that should I utilize my fellowship in some of the university and be with very ease start doing PhD there. So like nothing happened for some 15 to 20 days. While I was giving my ISER Mohali, I just passed the written test and I was sitting for interview outside the ISER Mohali auditorium. So what happened, I got a call. So this call was from the postdoc herself. She was Dr. Nirmala Jagdish. She said, Manisha, क्या कर रही हो आजकल?
So hello everyone, welcome to Bio Library, and after so long I'm recording this video, and I think your NII interview is approaching. ऐसे कितना सही.
Hello everyone, welcome to Bio Library, and let's start this video. As you might have guessed from the thumbnail, what guessed, you just know what the video is all about, right? It's about my NII interview experience. NII stands for National Institute of Immunology. Rest details you guys already know because you've qualified the exam, and just because you've qualified the exam, you want to know more about the interview process, okay? So I think you must have seen the interview of Zohra in my playlist. You can go there and check the PhD scholars interview column or the playlist, and you can just skim through her experience of NII, as she also gave NII interview, and mine was in the year 2019, and I think she recently gave this just last year. So it's always advisable that have multiple sources of people have multiple sources of information, such that you can get to know what all changes are there, or what all changes have been amended from 2019 to 2022. Okay, so first of all, very congratulations, heartily congratulations to everybody who has qualified the exam, and if you haven't done so, it's okay. How does it matter? Neither was I able to qualify the entire process. Mine is a different story. If you're following the series, my NII process or the NII interview or the NII PhD admission journey, then you must have known by now that I was not selected for PhD there, and I was called for a project which was NIH funded. So enough with my things. Like, I think you already know that I was not pitched for PhD admission, and one of the postdocs, ma'am, Dr. Nirmala Jagdish, she called me for this project vacancy. Okay, so let's start how my interview was. And like every other person, I was first anxious, second nervous, because I was coming from botany background, right? First, why did I went to such an institute which is completely devoted to animal sciences, or you can say is there's nothing for plant sciences there. So I went there first. It was my like responsibility to defend what my reasons were to choose NII. So I was nervous because I had to explain them my reason, and I had to make sure that they were also interested in knowing what my story is. Okay, so as I entered the room, first the entire interview committee was having some 4 to 5 members. To be very precise, there were 5 members, and they just I greeted them, and they were very cordial. They smiled throughout the process, and I, as you guys know, that always smile. So yeah, we were smiling throughout the process, and in parallel they were asking me questions. I was asked some 9 to 10 questions, out of which 2 or like there was a follow-up of questions from plant background, because I belong to plant background. Okay, so let's just divide it into 7 plus 3, 7 for the animal sciences or whatever is not there in botany, and 2 to 3 questions from plant sciences. Okay, so what I've done is I have done there were some questions which I needed to draw on board, and as Zohra, in her experience, you can learn that she gave online interview, so there was no scope of drawing anything or justifying yourself that whatever you're saying you might not be able to put it through words, but you know what you're saying. That's why you can go to the board, or they can suggest you to go to the board and explain them. Okay, so I did that in 2 questions, and those board questions I'll be drawing and showing you in the later part of the video. So I'll attach both the videos together, so I'll just compile them. So first, let's start with the first question. So first question is, they make you comfortable, that where you from and what were your educational qualifications till now, and were you comfortable while traveling to Delhi if you're coming from another city, as I went from Lucknow. Okay, so yeah, enough with the comfortable part. Then they started asking me a question. So the first question was asked by, there were two ma'am, three sir. So first question was asked by ma'am. She was sitting in, like, I can vividly remember that she was just sitting in the extreme left, and she asked me that, Manisha, okay fine, I am trying to express a heterologous protein in streptococcus, okay, but like in my consecutive 3 results, I have failed in getting a fairly high expression of protein. So she wanted to say that she's trying to express a protein. So heterologous protein is one. For example, it's my protein, I want to express it in yeast, I want to express it in E. coli. So expressing a protein which is of foreign nature, which is not innate in the organism itself, is a heterologous protein coming from different source. Let's put it in a simple term, that coming from different source. So she was saying that she's consecutively trying to express that protein, but she was not even getting a basal level or a high, or like some optimum amount of the protein. So what could be the reason behind that? So I think little did I understood about the question, but only I knew just one line of it, that she's trying to express heterologous protein. So what could be the problem? Like, the problem is in the system she's using, or the problem is in the gene? Why is it not getting expressed? So I asked some of the questions like, ma'am, can you please elaborate? So she just break down her question, which gave me a hint that okay, so it's about the system. Okay, so I said, ma'am, I think it is because of codon bias. So she said, like, when people encourage you, she said, okay, good, can you explain more about codon bias? So I said, ma'am, there is some problem in the system, that the codons which are required for that heterologous protein are very rare in the system. Therefore, there are not enough tRNAs for it, which is what codon bias is. I was just putting it in my words. Like, I was not this much confident. Right now I'm telling you. So there I was, a little, random in my answer. So I said, ma'am, we are trying to express it. The amino acids for the heterologous protein are very rare. Therefore, there is not enough of tRNA present. So therefore, the system is not giving us the optimum amount of protein. So then she said, okay, if I take you in my team, what would you do? What would Manisha do in this situation? How will you rectify this problem? So I said, ma'am, I will impart the system enough of tRNAs. So in that case, what you have to do is, you have to take a plasmid, you have to clone tRNA, you have to provide the system enough of the tRNA such that it gets charged with the rare amino acids, like the tRNAs, and therefore your system start forming, because it does not have enough of the codons. The codons were rare for that amino acid. Okay, so this was my answer, and I found it her like she was nodding, so I thought, okay fine. I kind of approached the question, so that boosted a little bit of confidence in me. What happens is, after some time your default state gets expressed. If you're an extrovert, after some time you'll be very comfortable, and you'll be all your energy will be in giving the answer. But if you are an introvert, here comes the problem, that you will start thinking about yourself. So whatever your default state is, try to practice in front of the mirror. Okay, fine. So the second question came as, can you differentiate between cloning vector and expression vector? So I said, if I want to express a protein, if I want to express a gene per se, so I will use an expression vector, otherwise I'll use cloning vector. So she said, okay, go to the board and draw it. So I'll show that in the subsequent segment of this video itself, but I'll tell you here orally what did I say. So I said, ma'am, it will have some important components like I draw there, but what I did wrong there was, I didn't draw RBS. Yes, I didn't draw RBS. She said, Manisha, are you forgetting something very integral? I said, okay, so I have to express the protein, then what do I need? So I said RBS. So she was like, okay fine, she knows the basics. That's what they're trying to check, that are you thorough with the basics, because nobody's checking you. You're not a doctorate by yourself, right? You have to do it. So you need to be very much thorough with your basics and very much confident in your basics as well. So okay, fine. So RBS wala scene happened there, and then one of the professor, second or third, he asked me that okay, now what you have to do it, you make it a system which is like I can regulate the system. You have to make it a system such that it can be regulated. So little did I know, again it was like whenever any regulatory system comes in mind, operon comes in mind. Okay, so I thought maybe I could insert some of the regulatory element there. Then I tried to draw the regulatory element. She said okay, you're doing good, but you don't you think you should draw a regulatory gene there? You can't give the regulatory protein itself. So I said okay ma'am, so I can do one thing, regulatory gene insert कर देते हैं. What will happen is उसका जो product होगा it will bind to the operator, and according to the requirement, according to the system, whether catabolic anabolic, वो it will become indicible or repressible. Okay, so what happens is you can't judge there, whether you're going right or wrong. By their expression only you can be confident or be calm, I would say. So all of them, like, they were smiling. So what I realized was they were interested. Till this question they were interested because everybody was listening to me. Not even a single person was doing this and that, which also happened with me. Like in the earlier case, when I'll tell you about IIT Roorkee interview experience, then I'll let you know what happened there. So there were those in IIT Roorkee, there was so much of your neck rotation. There were some 15 to 16 people sitting there. Okay, fine, I'll leave it. Then she, I think ma'am only told me, or sir, which I'm forgetting, which is like of no use here. So someone from the faculty said that okay, draw or go to the board and write. I was standing to the board itself na. So they said ki okay, go and write the constitutive cases of lacoperon, like your system is constitutive. So I did that. Again, I'll draw it later. And then the questions came from, okay, till now we have dealt these questions. And then yeah, one of the professors said, Manisha, you belong to plant background. Okay, so what will you do here? Why are you coming to here? Why are you coming to NII? That's what I said. I mean, I was thinking, are they interested in my story? So here my story begins. I said ki sir, we can do some work in collaboration with NII PGR. I was so childish, you would say. Okay, it's fine, but I expressed myself. I said, sir, we can collaborate with NII PGR and we can utilize some plant-based products in curing this cancer problem. So I was so adamant on working on cancer. So okay, they said okay, fine, if you want to work on cancer, then please tell me what are the medicines extracted from plant which are anti-cancers. So of course we all know that vinca rosa. So from vinca rosa we get vincristine, vinblastine. Okay, then comes from this question, what is the function of colchicine, what are the respective functions of vincristine, vinblastine? That I know, of course you guys also know. So what was their question now, which was like I was taken by surprise. They said, one of the faculty was like, okay Manisha, let's see how observant you are, right? How present in the moment you are. So he said, okay, so you came from the main door, can you please tell me how many vinca rosa have you spotted? So I said, sir, there are many, because see, I knew just one thing, it's sadabahar na, it is always like flowering. So I said, sir, there are many. And he said, was there any flower? So I said, sir, some of them were, yes, flowering. So they started laughing. I mean, whatever happened, I think I had seen some 1 or 2 catharanthus there, Catharanthus roseus, which is vinca rosa. So I said that. I mean, I was not hesitant, because in this case, what they could guess is this girl is observant, non-observant. So whatever the conclusion might be, the conclusion can also be that the child is nervous, right? So don't get intimidated by such questions. They can ask you anything. Like in BRC, it was asked some question for wheat production, wheat market in Lucknow was asked to me. So okay, let's jump to the next question. So the next question was, do you know about immunoelectrophoresis? So here I want to pitch you an advice that do one thing, read about all the immunotechniques. Okay, you're going to an institute which is concerned with immunology. So it's very cliche that people will say, why will you read immunology? I ask you, why won't you? Right, first thing you should do is read immunology. Go read immunology. Immunotechniques are very important. and then you'll be confident enough that you know the basic, right? You know the basic of the institute. Okay, fine. So they asked me about immunoelectrophoresis. So I think don't be, very confirmed of the fact that she knows everything. I didn't know everything. It happened because I gave BRC interview just 2 months, just 1 month or some days back. I think before this NII interview, and after there I was asked about immunoelectrophoresis. What happened? They were asking about some modification of electrophoresis, and I just uttered this word immunoelectrophoresis, and there was a ma'am. She started laughing that Manisha, does something or anything in this world is like immunoelectrophoresis? Does it exist? So I said no, ma'am, because in BRC I was not confident that immunoelectrophoresis, does it exist or not. Then I came back, I read on Google, and I get to know that okay, fine, there is a technique called immunoelectrophoresis. When you have a lot of antigen, when you have a complex mixture of antigen, you can separate them with the help of immunoelectrophoresis. It's basically the, what do we study? Octoloni diffusion method, that is diffusion plus electrophoresis, diffusion of antigens plus electrophoresis, and then antibodies are applied to it respectively. Okay, so also there are advancements of this immunoelectrophoresis technique, like affinity immunoelectrophoresis, and there are some 2 or 3 more. So there is a technique for immunoelectrophoresis. Please do one thing, just read your either Pathfinder book, Wilson Walker, T.A. Brown, whatever you are preferring for techniques. Read the immuno techniques thoroughly, and then just one thing, go to Wikipedia and read what all are the advancements of the techniques and what are they getting used for. See, techniques is very, what do I say, it's very easy when you just start giving 15 minutes of every day to techniques, because you have to become a PhD scholar and you need to develop your scientific temperament. What happens after reading from Google is you get some 2 to 3 more related questions by which your knowledge enhances. Okay, fine, this was also there, okay, this was also there. Whereas in your book the information is static. Okay, you've read till that point, you have made your basics very clear. Go to Google, go to Wikipedia, check about the variables, and also type immunotechniques.pdf. You'll get some of the research papers on this immunotechniques. Okay, and on immunoelectrophoresis you can read one research paper which was published in 2009. I cannot assure that the same question can be asked to you, but yes, it can be asked. There is 50% chance. Okay, so yeah, this happened. Immunoelectrophoresis, I gave answer. And then I was asked about that, have you done any project in your masters? Because I was without any file. Like, see, everybody knows she hasn't done anything, therefore she is without any file or something like that. Okay, so I said no sir. Then they asked why. I said because in botany department of Lucknow University they don't allow us, or there's no provision of doing project as in wet lab or something. So they said, then what do you do in your 4th semester? I mean, of course, a natural question, right? So I said, sir, we do book review. Then they asked that, see, sometimes they can be very intimidating. They were like, what were you doing in your 4th semester then? It happens, so don't get intimidated by the tone. They are elderly people. Okay, so it happens. Some of them might be acting to scare you. They just love it, because I also think when I'll become a committee member, I'll also do the same. Okay, so yeah, so what happened is they asked me in this manner, and I said, sir, we do book review, and mine was on tissue culture. Then you guys know what questions can be made from tissue culture. I was asked a question on what will happen when auxin will be more than cytokinin in your tissue culture media. What are somaclonal variations? What do you understand of inbreeding depression? What can you infer from the idea of this inbreeding depression and vigor, hybrid vigor? All these are botany related terms. So they asked me very general questions. And also one of the members said that, Manisha, what if you are fooling us? I said, I didn't say anything. I just smiled innocently. They said, Manisha, we all are from animal background. So do you want us to call some person from NII Ph.D to just cross check what you're saying? I said, yes, sir. So they called some of the professor, and he said he'll be there in 10 to 15 minutes. But I think they didn't have that much time. So they said, okay, he'll be late. Then what should we do? So I said, sir, please reconfirm me with Google. So they said, okay, fine. So they started laughing. Okay, so do one thing. If you don't know the answer, and just don't allow your ego to take place that, sir, how can I fool you? Why will I fool you? Don't do that. That's a victim mentality, of course. You'll be like, okay, sir, I mean, just go with the flow. Okay, interview mates more about your personality, what you are as a person. You can fake a person under pressured situation. You'll be in your default state in one or the other form. Okay, they'll do that for you, to you. Okay, chalo. Then the next question. Next question again came from sir. This was the person who again tried to contact me for cancer research, and he asked me, okay, you want to work on cancer, and I'm working on one of the project, just this much. He said, okay, do you know about ETBR? I said yes, it is ethidium bromide. So he said, okay, so can you please tell me that why does it fluoresce more when it gets intercalated as compared to when it is poured in the gel? So I didn't know anything about this. I've never seen this, but I told the person that, sir, will you please simplify the question for me? I would like to attempt the question. So you'll say, Manisha, you're very frank that you say everything to the professor that, sir, I would like to attempt that question. See, what happened with me that I gave interview in BARC for 1 hour and 15 minutes. Before that, I was in, I went for interview to IIT Roorkee. I gave interview, I think, was Isa Mohali done? No, Isa Mohali was left till then. And I guess also NII PGR was left. Like, I think I have had enough experience. I gave PGI before it, and I gave BRC before it. I gave IIT Roorkee, and I'm an extrovert for that matter. I love oral presentations. I love speaking. I think I love being under pressured situations, because I know what will happen. I mean, they won't take me as a student. Okay, I mean, how does that matter? So yeah, okay, fine. That's why I said that person that I would like to, attempt that question. Can you please elaborate it for me? So he said that, Misha, whenever ETBR is there, when you all know that DNA is visualized with the help of ETBR. So what happens is ETBR intercalates in the DNA, that you know. Okay, fine. Then what happens when it is placed in the buffer, when it is placed with the contact of the buffer, then it does not fluoresce that much as compared to when it comes in the contact of DNA, it starts fluorescing at a very high intensity. So see, I just took one hint from here that there is happening something to ETBR which is restricting it from, fluorescing there. So I had the idea of quencher. From earlier, like I read Frit, I read some of the phenomena, and I had this idea. I said, sir, it is getting quenched. So he said, okay, fine. He smiled, and I was also very happy that I could, attempt the question. I could never ever attempt the question if you've asked me before that situation, because the person elaborated the question to me in such a manner that I thought, let's go with quencher. Okay, so then he said, so what do you think? What is the component which is quenching it? So then again he said, it is placed in buffer. So I said, sir, one of the component of the buffer. So he said, what could be there in buffer, like in the highest concentration? I thought solvent, right? I said buffer is itself a solvent. So like one or the other form, there must be distilled water, double distilled water in it. So I said, sir, is it water? I'm not very sure, sir, is it water? So he said, yes, it is water, and it is getting quenched. Therefore, it does not fluoresce with the same intensity as it does with the DNA, because it leaves water, it enters the hydrophobic environment, and it fluoresces at a very high intense rate. That picture I will show you here. I read a research paper after that, because I was asked this question in a very casual manner by one of my seniors, but I was very happy that I knew it from earlier time. Okay, so one more question from ETBR came from one of the scientists. And what happens is, whenever somebody gives you one question, then there is some professor thinking some of the modification of that question only. So they said that why can't you visualize single stranded DNA with the help of ETBR? So I didn't expect this much simple question after this ETBR question. I was in so much of pressured situation. I was like, okay, fine, ab to jang jeet liye kind of situation happened. So I said, because it intercalates between double stranded DNA, therefore I can't see single stranded DNA with it. So yeah, that was about it. That was my interview. And then they said thank you, and then again this professor who asked me this ETBR quencher wala question, he said that so you want to work in cancer. See, these are hints that the other person remembers you or is trying to remember you. Okay, so Then they said, okay, thank you, and I left the place, and I was expecting my name to be in the list for second round of interview, but it wasn't there. It was shattering, of course, but I then make myself understood that you yourself have gone to Animal Sciences Institute saying that I would like to collaborate with plant sciences. Who are you? Like, and why would they take you? So yeah, I thought I'm not good enough. Like, I used to criticize myself back then, so I thought, okay, fine, I'm not good enough, therefore they haven't taken me. So yeah, again, this cycle of, judging yourself, criticizing yourself, and thinking that you're not good for anything, and you are just roaming from here and there. All that happens, and you just think in your back that, why should I utilize my fellowship in some of the university and be with very ease start doing PhD there? So like, nothing happened for some 15 to 20 days. While I was giving my Aizer Mohali, I just passed the written test, and I was sitting for interview outside the Aizer Mohali auditorium. So what happened, I got a call. So this call was from the postdoc herself. She was Dr. Nirmala Jagdish. She said, Manisha, क्या कर रही हो आजकल? So I was like, I didn't say anything. Then she said कि there is some project vacancy, and sir has said that he has released it specially for you because you wanted to work on cancer, right? So if after 1 year your performance would be satisfactory, then he would like to take you. He will enroll you to PhD position. So what happens is, at that time you don't have that much idea as to what project is. Why should I do project when I am already a JRF? Should I go for PhD positions? And there is nobody who is telling you this, who is telling you that is a project good option. There's nothing like that. I don't think it is still there. So yeah, then she said this much, and I in my ignorance said that, ma'am, mera Aizer Mohali ka written nikal gaya and I'm waiting for my interview. So it was arrogance. It was just that because she said, Manisha, खाली बैठोगी सारे साल, sorry, पूरे के पूरे साल खाली बैठोगी क्या? तो I thought, let me tell her that, हम भी champion kind of है. Okay, fine. Well, I thought, let's just tell her that project क्यों करेंगे हम. So that was the kind of mentality I have had, and I would love to tell you why should you do project, what are the merits, demerits, and should after JRF is doing project a good option for you or not. That I would love to tell you, because I have a lot of anecdotes to share. I have a lot of examples to quote, mine like itself, including mine as well. So enough with this thing. I'll add one video where I'll explain these two questions. One is the expression vector. I know you guys know, but I would like to tell you how to draw it when you do it. So just draw one circle first whenever anybody says you to draw any vector, okay? So that vector could be linear as well. Ask yourself, that vector could be linear as well. Okay, fine. So I'll tell you that after drawing on a piece of paper, that how I went to the board and draw that for them. And there are 3 takeaways, guys, which I would love to tell you. That when stuck in a situation of a PhD interview, what should you do? Okay, so the first being, just do not stop pushing yourself. Just keep pushing yourself. Like I quoted the example of quencher wala question. What happened there? If I'd never seen ETBR in my life. So how did I answer? I answered because I kept on pushing myself that, have I heard about this situation before? Did I know anything? So I just kept on pushing myself, and I got the answer. So you will end up making miracles for yourself if you will keep on pushing yourself in the pressured situation. Comfort is nothing. There's nothing like comfort exists in the pressured and fearful situations. Okay, so you have to go for it. Completely go for it. Second one is, if you in your head think that there is a 50% probability that what I'm thinking is wrong, that please allow me to impregnate that part of your brain where you're thinking that the 50% of what you're thinking is right? If it is right, then you should go for it. Just say it. What will happen? They are not gonna beat you, right? What else? So just don't be arrogant in a very polite manner. Just tell it, okay, fine, is it like this, is it like that? So you can do that. And the third one is, just in between by their expressions, do not judge that whether you're getting selected or not selected. That is none of your business. Whether or not you're getting selected or not. What your business is to give your 100%, to tell them that this 15 to 16 years of education have made you this person, that you can handle this situation gracefully, as well as if you're getting failed here, you can go gracefully as well and do well in your life. Okay, so you don't have to get demotivated and start crying, because I did that in my previous one of the interview. I did that also. So don't do anything like that. No, your life doesn't end there, right? So after some time you laugh on those anecdotes, but do one thing: be very peaceful there, be very calm, because you don't get interviews anytime. Like it's not like you have to be a JRF for it, you have to qualify the exam for it. So you have something in you, therefore you are sitting there. Okay, so there are some scientists who have gone through the wonderful journey of PhD, postdoc, and then fighting for PhD, fighting for faculty positions, and then they are sitting there. So you are getting to interact with the most wonderful and the most intellectual minds of India. So please be very positive about it. Whether or not you qualify the interview, none of your business. Again, I'm telling you, just keep on telling it to yourself that I have to give my 100%, beyond 100% if required. Okay, so with this, I'll take your leave. I'll add that video here, and thank you everybody. I think you guys are awesome and you're gonna make it someday or the other. Okay, so bye bye. And my first question was, just go to the board and draw expression vector. Okay, so you all also know that what all are required for expression vector. Just you have to keep in mind that an expression vector will do what? Will help you in transcription as well as translation. Like you need to express your gene there. So first you'll have to make the RNA component and then the protein component. Okay, so what I did was I went to the board and then I made a circle, as I told you. And then this is your question, that you have to think that what if you are the vector is linear. So it's your question, can it be possible? So first I did what? I made origin, and then I made your selectable marker. I'm typing SM here, and then here what did I do? I made your here and here. From here this is your terminator sequence. Okay, so this is your promoter. So what happened? Promoter was here, terminator were here, and then what I required was your coding region. Some of the region was your coding region, right? And then I also did one thing, that I made a tag here. I said, ma'am, this can be a tag such that if you want to purify your protein, CT tag bhi ho sakta hai, C terminal tag can it also be. So it can also be C terminal tag, because if you want to you want to purify and collect your protein later, so this tag will be helpful there. Then what happened? She asked me that, are you forgetting something? Then therefore you can also see that this is incomplete without what? This is incomplete without RBS, right? So I remembered, that was fortunately I remembered that what I was forgetting is RBS. So I made sure that I wrote RBS then. And then one of the faculty, as I told you earlier, asked me that make it a controllable or regulatable system. So what I did was, I just said that I'll add some regulatory element to it, like the coded regulatory element. Then they told me that why don't you add regulatory gene to it? Then I typed that I'll add a regulatory gene to it, such that its product, whatever I gene, for example, whatever its product will bind here to the operator region, and then will control the system in the required manner, such that it can be what? Inducible. It can be inducible or repressible system. So what was what was asked to me? It was asked that please draw an expression vector. So what did I do? I just in my mind I revised that what all are required to be a expression vector. So transcription, translation, uski essential features hai. And then what I did was, I started by making a circle. Then first I draw this origin of replication, then selectable marker. Start with the essential things: promoter, terminator, of course required, and then I draw this coding region. As this C terminal tag, I am very proud of that I made it, because I didn't know that why is it used. And then I later, when she corrected me, and then I draw this RBS, and then the question of regulatory gene came. Okay, so about second question, the second question was, write the what? Constitutive cases. I think you guys already know what are the constitutive cases. What are the mutation, like your merozygotic cases? What are the merozygotic cases in which your system becomes constitutive? It is always on. Your system is what? it is constitutive. Okay, so it will always be on. So as we all know, there are only 3 cases for it. So what can happen is 2 mutations in your operators, 1 mutation in your operator, that is constitutive mutation in your operator, and the second mutation is in your repressor. Okay, so either the repressor is completely non-functional, or repressor is not made, it is not synthesized, or the second one is a repressor with a mutation in the DNA binding domain. Okay, so these two mutation, one mutation is this, then this second mutation is it cannot bind to the, so its DNA binding domain is defective, so it cannot bind to your this region. And then the third or the most important mutation is operator, the constitutive mutation of operator. So these are the 3 cases in which there will be a constitutive expression of your lac operon, that is the system will remain always open. Okay, so with this and all the takeaways, you just remember all the takeaways I told you earlier, and all the best for your interview. I know you're gonna do it and you're gonna do very well in your interview, not only NII interview but in all the rest upcoming exams of your life. Okay, so everybody, thank you and keep studying, and please tell me how my efforts are helping you. Okay, so anything, if you think you want help, you can type in the comment section. So thank you everybody, we'll meet with the next video or next interview experience of mine. Okay, so keep studying.